Compound microscope. Objective: to study the construction and working of a compound microscope. We have already studied that a magnifying lens is a simple microscope, which is used to magnify small objects into big ones. Just imagine how exciting it would be if we get a device in which we can clearly see the minute objects like cells, which cannot be seen with the naked eye. Well, that is the magic done by a compound microscope. A simple magnifier has limited magnification, but a compound microscope has much more magnification. Now, let us study the compound microscope in detail. Let us see the external construction of a microscope. The compound microscope consists of an eyepiece through which we see the image of the specimen. Then is the body tube, which is a long tube holding the eyepiece and connecting it to the objective. This is the revolving nose piece, which holds the objective and is the rotating part of the microscope. Then we have the objective lenses, which are attached to the nose piece. The arm is the part of the microscope with which you can hold the microscope. Then we have the coarse adjustment knob, which is a large round knob on the side of the microscope and is used for focusing the specimen. Adjacently, we have the fine adjustment knob, which is a small round knob on the side. Next, we have the stage, which is a large flat area under the objectives, on which we keep the specimen. Stage clips are used to hold the slide in place. At the base of the microscope, we have a mirror, which focuses the light onto the bottom of the stage. Some microscopes use a light source to produce light. The stage has an aperture, which is a hole that allows the light to pass through the specimen for viewing it better. This is the diaphragm, which controls the amount of light going through the aperture. In its simplest form, the compound microscope would have a single glass lens of short focal length and small aperture for the objective. And another single glass lens of comparatively larger focal length and larger aperture for the eyepiece or ocular. Next, let us see how all these parts work together to give us the final image. Let AB be an object placed just outside the focal length f0 of the objective lens. Here we can see a real inverted and magnified image, A-B- is produced on the other side of an objective lens. Now, the separation between the objective lens and the eyepiece is adjusted in such a way that image A-B- falls within the focal length FE of the eyepiece, as you can see here. Now, the image A-B- acts as an object for the eyepiece. Here, the eyepiece will act like a simple microscope or magnifier, and hence it forms a final image, A-B-, which is virtual and magnified, and is formed on the same side of the eyepiece lens. We see the clearest magnified image when it is formed at the least distance of distinct vision D. In compound microscope, distance between the eyepiece and objective lens is known as tube length L.